Hi, welcome back to the Sound of Science AP edition. Today we're going to be talking about enzymes. This covers AP Biology Units 3.1 through 3.4. As you may remember, enzymes are macromolecules. Specifically, most of them are going to be proteins, although there are some enzymes that are RNA based, which is actually kind of cool. Enzymes work as biological catalysts. They speed up biochemical processes. Proteins have a tertiary shape that makes it functional. So if you change the shape, you inhibit the protein or inhibit the enzyme. This is what we call the lock and key model, and I have a picture of below. The enzyme itself is going to have an active site. That's where whatever molecule it's bonding to, the substrate, can bind to it. And then it can cause a chemical change, or sometimes not. Sometimes it causes a physical change. But regardless, the size and the shape matter. Otherwise, the enzyme and the substrate can't fit together to create the enzyme substrate complex. When you look at the lock and key model, one of the things that you need to keep in mind is that the physical and chemical properties of the substrate have to be compatible with the enzyme. There may be some slight changes in the enzyme that help the substrate and enzyme align together, but for the most part, there's not going to be any differences in the enzymes. Changes can lead to it becoming non-functional. Enzyme names usually end in ASE or ACE. So, for instance, lactase, digest, lactose. Enzymes are reusable. They're not going to be chemically changed by any of the reactions that take place, and they're not going to be used up. So, they can actually be used over and over again. Enzymes, however, are very, very specific. They can only be used to facilitate one type of reaction, and each enzyme only fits very few molecules. So often the reactions that are taking place are synthesis or digestion reactions and only of the specific item. Typically, they're going to be named after whatever they're digesting or synthesizing. So, in the case of sucrose, sucrose is going to be digested by the enzyme called sucrase, and it's going to turn it into glucose and fructose. This diagram seems really complicated. It's not as complicated as it seems. So, this is talking about activation energy. You, in every reaction that takes place, need to supply some type of energy to get the reaction going. Okay. All biological reactions require some sort of starting energy, and we call that activation energy. Reactions can either result in a net release of energy or a net absorption of energy. If there is a release of energy at the end, usually we don't need as much activation energy to start with, which kind of makes sense. Enzymes reduce the amount of activation energy that you need, making that reaction happen faster. So, in this case, right here, what we're looking at, so our reactants are glucose and oxygen, right? And we have released energy and we have energy supplied. So, you see that without the enzyme, we require quite a bit of energy to get the reaction going. But once it starts going, it ends up producing energy for us, right? And the products here are carbon dioxide and water. For those of you who didn't catch on already, this is actually what we're talking about. This reaction is actually cellular respiration. So without the enzyme, we need a lot more energy than with the enzyme. With an enzyme, this reaction actually takes place with a lot less energy, ending up in the same place with the same amount of energy being created. That's why these enzymes are so important. Enzyme activity can be affected by the environment in which it finds itself. And in fact, because the specific shape is so important to enzymatic function, that any type of temperature change or pH change can actually lead to what we call the denaturation of the protein or of the enzyme. Denaturation is what we call it when the enzyme shape or structure is changed to the point that it reduces or prevents the enzyme from functioning appropriately or correctly. That shape, by the way, of the enzyme, we call that the conformational shape or the tertiary structure. So make sure you understand what those words are. So if you look at the photos or the pictures that I have at the bottom, you can see the shape of this protein right here. And that is a beautifully folded protein. It has several amino acid chains sort of stuck together. They're all folded in a specific way. They're held together with some hydrogen bonds. Everything looks good. This is the shape it needs to be. This is a globular protein. Adding heat, in fact, too much heat, can actually cause this protein to start to break down. It actually unravels it, prevents it from keeping its shape. What this would look like in, for instance, some item, take these eggs, for instance. The clear part of the egg, this is what's going to become the egg white. This, once we add heat, turns white. This is because we're changing the protein structure. When it comes to the temperature in the environment, the optimum temperature is the range at which the enzyme functions the best. Okay. So if we change the temperature, we're going to change the rate of the reaction. For instance, if we increase the temperature of the environment, typically, initially, we're going to see an increased reaction rate. And that's just because the enzyme and substrate are going to collide with one another more often. That increases the likelihood of the reaction occurring. However, if it gets too hot, the enzyme itself is going to denature. And think of that egg white turning white because of the heat. Decreased temperature is going to decrease the reaction rate. Again, it decreases the frequency of the enzyme substrate collisions, but typically this is not going to lead to denaturation. And if we increase the temperature again, we can actually bring up the rate of reaction again. pH is a measure of the concentration of hydrogen ions in a solution. And typically we think of, like, if we have a pH of less than 7, then we're looking at something that's probably acidic. If we have a pH that's more than 7, we're looking at something that's pretty basic, right? That's how we typically think of it. But really, that scale is logarithmic, which just means that a pH of 6 actually has 10 times more hydrogen ions than a pH of 7. So a small change in the pH number can actually cause big differences in the amount of hydrogen ions in solution. The optimum pH is the range at which each enzyme works best. And again, this is going to be different. It's going to be dependent on the enzymes. Some enzymes function well in an acidic environment. Those hydrolytic enzymes, the ones that are found in lysosomes in our cells, actually function really well in an acidic environment. That's why they have to be contained. Increasing or decreasing the pH, though, can cause denaturation. And it's really because it's going to affect the hydrogen bond interactions that are occurring, changing the shape of our molecule. The concentration of substrates and products. So if we add lots and lots and lots of substrate, that raw material that we're trying to bind to the enzyme, it initially is going to increase the reaction rate, but eventually it's going to plateau at the substrate saturation. That's when all enzymes are currently being used. It doesn't go beyond that point. So how best to think of that? Think of, like, you go to Walmart and they have 5 checkouts open, right? 5 registers are open. That means that Walmart can currently check out 5 people. That's it. They can check out 5 customers. That's it. That's their max, because that's all they have. Now, if we look at increased concentration of product, that actually is going to lead to a decreased reaction rate pretty quickly. The products take up space and they actually prevent the substrate complex binding from occurring. This would be if you were trying to check out at Walmart and it was Black Friday and there's people everywhere and you can't even get to the register. Now, let's say that we look at enzyme concentration. If you have less enzyme, you have a slower reaction rate. However, if you have unlimited enzyme and unlimited substrate, the reaction rate is going to continue to increase until you run out of one or the other. You're going to find it continuing to go. The last thing that I want to talk about today are inhibitors, and there's actually two types. We have competitive and non-competitive. I'm going to start with competitive inhibitors. Basically, a competitive inhibitor is going to bind to an enzyme's active site, even though it is not the substrate that typically would bind there. It's another molecule that's come in and it binds there for whatever reason. It happens to fit. It's chemically compatible. So it binds there and it prevents the substrate from binding to the active site. This means that it's going to slow the reaction rate because the reaction can't take place if there's no enzyme there to allow that to happen. If you have small amounts of inhibitor, the enzyme and substrate reactions typically are going to stay at a pretty normal level. If the inhibitor, however, binds permanently, that means once it binds to the active site, it's there, nothing's going to kick it off, then the enzyme's function itself can be prevented forever or indefinitely. So something to definitely kind of keep in mind, these competitive inhibitors just basically block it. They keep things from happening. So what good would that do? Well, actually quite a bit of good. Penicillin is basically a competitive inhibitor that is going to bind to some of the molecules that are found in bacterial cell walls. It's non-reversible. That means once it binds, it's there. And this actually is what's going to tear apart bacterial cell walls and kill the bacteria. That's why antibiotics work the way that they do. Aspirin, or acetylsalic acid is its other name, is also non-reversible. It inhibits a specific enzyme from functioning, and this actually helps prevent inflammation and helps prevent pain as long as the acetylsalic acid is in the system. Once the acetylsalic acid starts to fade, it starts to break down, then that pain and inflammation will start to return. So we actually have lots of different medical benefits that can come with this type of inhibitor. The non-competitive inhibitor binds to what we refer to as the allosteric site rather than the active site. This causes changes to the shape of the enzyme so that it will no longer function. Imagine that you have a balloon and you squeeze one part of the balloon. It's going to change the shape of the rest of the balloon. This means that perhaps our substrate can't bind to the enzyme because it's changed shape, which is what you're seeing here in these pictures. Increasing the substrate's not going to prevent that. We can't add more substrate and get our levels back up because these enzymes just simply won't function. Their shape's been changed and they can't do their job anymore. So that is all the time we have for right now. As always, I will see you next time on The Sound of Science.